Good afternoon, everyone. Welcome back for the afternoon session. My name is Wenbo Li. I'm a editor from Nature Communications, handling for structural biology, biochemistry, and biophysics. My remit is on all structure-based protein and machine learning-based structure prediction, protein-protein interactions, and everything structure-based. So in this afternoon's session, there will be four keynote talks, and 2 flash talks. So the first speaker is Professor Rama Raghunathan. Rama is a professor in the Department of Biochemistry and Molecular Biology at the University of Chicago. He is also the director of BioCar's Beamline, a synchrotron facility for structural biology at the Argonne National Laboratory. Rama grows up in San Diego, California, and received his medical degrees and PhD from University of California, and postdoc work was carried out at Harvard Medical School. He was a faculty member at the University of Texas Southwestern Medical Center for 20 years before joining the University of Chicago in 2017. Rama's work focuses on understanding how revolution works as a design process to produce the remarkable high-performance adaptive machines that makes life possible. In this talk, slightly different from previous versions, this is a recorded talk, Rama will discuss the key steps towards the theory of physics of proteins that is consistent with evolution.
Well, it's a great pleasure to give a talk at this meeting. I want to begin by thanking the organizers, as well as our colleagues at Nanjing University for inviting me to give it. And so let me tell you about this problem that we've been working on. We call the evolutionary design of proteins. So we're going to spend the time today really talking about the principles of protein engineering and design. But I'd like to start with a bit of a more general overview. The interest in our group really lies in this problem. Biology is made up of these large nonlinear dynamical systems that operate from the scale of atoms, proteins and other molecules, all the way to the scale of ecosystems. And it's been a major goal for all of us in biology to understand their design, that is how they work, and of course also their origin through the process of evolution, because it's those problems that ultimately hold the key to a more abstract understanding of biology, and indeed hold the key to a future of engineering these systems. The difficulty, of course, is that these are really apparently very, very complex systems. So let me just show you what I mean by this concept of biological complexity. So this is a portion of core metabolism in bacteria, actually, and in some sense, this is a triumph of human progress over the last 70 years to work out all the parts and reactions in these very highly complex systems. But if you just step back and ask a question like, what is being computed here? How are inputs into this network converted into outputs? It's been very hard for us to answer that question because the system is made out of a lot of interacting parts, and a lot of those interactions are nonlinear. So it's been quite hard to understand these sort of systems. What is changing the world significantly in the recent history is a thing that's going to be very familiar to people that are attending this audience, which is this, what I call the statistics before physics approach. The idea that we're able to generate very large data sets across the hierarchical scales of biology. And the question is, can we make low dimensional representations from this data, models that give us the rules of understanding and rules of design of these systems. And so, as you all know, this sort of statistical rather than mechanism first approach is really fundamentally changing our approach to biology. And let me tell you the story of our work in that area in the scale of proteins. So, you all know that proteins are these nanoscale machines that occur in cells that carry out essentially all the chemical reactions necessary for life. And again, it's been a dream to understand their design for both, fundamental reasons, how evolution builds these large complex collections of atoms, and of course, for practical reasons of solving a lot of important problems in the world. So how is the statistical approach sort of borne out in the scale of proteins? Well, this is a sort of an old story. I mean, this is work that began really 25 years ago and here's the initial ideas that, if you want to understand the principles of design of a particular protein, let's say this one on the left, the idea is to not go and do direct physical measurements and experiments on a single instance of this protein. Instead, you should go get yourself a large and diverse representation of sequences that represent members of that protein family, homologs that share a common structure and common aspects of function. And then you should pose the problem statistically. Look at the frequency distribution of amino acids at positions in that protein, and also look at the second order terms, the correlation of outcome evolutionarily at one position with another position in the protein. And these sort of approaches lead ultimately to data sets that look like this matrix on the right. So this matrix tells you how every position in this protein going from N terminus to C terminus is correlated with every other position going from N terminus to C terminus. So this is a diagonally symmetric matrix where the diagonal terms tell you something about the intrinsic constraints at that position in the protein, and the off-diagonal terms tell you how one position in the protein is correlated in evolution with some other position in the protein. So in some sense, seen through the lens of evolution, how all the amino acid positions in a protein talk to each other. Okay. Now, a lot has been done in interpreting these sort of matrices of amino acid interactions seen through the lens of evolution about how proteins work. How are the reaction coordinates constructed? How are proteins able to fold? And how are they able to evolve, actually? But I'm not going to tell you any of those things today. I'm going to actually focus on a particular aspect of this information, which is to say that these are really low-dimensional, simple models of proteins. I mean, after all, in constructing this matrix, we're only extracting the first-order and second-order statistics of these sequence alignments. We're ignoring, for example, 3-body and 4-body and higher-order terms. And remember that all this inference of these statistics is made in the limit of extremely bad sampling of sequences. I mean, we may have 1,000 to tens of thousands of examples of a particular protein family, much less than needed even to constrain the pairwise terms. And so these matrices are in some sense noisy representations of evolutionary interactions of amino acids. So it's really an important question to ask, how much information about proteins do they really capture? And the best way to address that question is through protein design. The idea being that whatever you learned in these low-dimensional models, contaminated by whatever noise and limitations, if you say that they are really sufficient to represent a protein, well, the proof lies in designing synthetic proteins from the model using no other information, and go to the lab and build these things and ask, are they truly synthetic homologs of a protein family? And here are the results of such studies now quite long ago, actually. So here I'm showing you data for 2 different protein families, the WW domain proteins and the PDZ proteins. On the left are examples of what nature made. So this is the human PIN1 WW domain, and here's a rat brain PSD95 PDZ domain. On the right are actually experimental structures of synthetic proteins made using no information about the structure, and indeed no physics at all, just to satisfy the pattern of pairwise correlations that we learned from models like I showed you in the previous slides. And the bottom line is, with that kind of sequence identity and that kind of probability of folding, we're able to make protein structures that are essentially indistinguishable from what nature has made in those protein families. So this kind of work really established that these sort of small set of numbers you learn from just even the pairwise coevolution terms are sufficient to rebuild a protein fold with extraordinary sequence divergence. But the interesting result is it's not even just about folding. These proteins actually work, and they work in the same way as their natural counterparts. So WW domains are polyproline binding motifs, and particular members of the WW family show specificity for various polyproline motif. So for example, this particular one, this one here, binds PPXY-like motifs. And here on the right, you can see that the synthetic protein actually shows the binding specificity that mirrors the natural protein. This is a legitimate class 1 WW domain, and both the specificity and the affinity are the same as the natural protein. And here on the bottom, I'm showing you that the synthetic PDZ domain in red binds a particular C-terminal sequence in the mouse genome, which is in the same range in affinity as all the mouse domains that bind that same C-terminal peptide. So, these data already started to show us that both folding and specific aspects of biochemical function are encoded in a very, very low-dimensional and say simple set of simple representation with a relatively small set of numbers. But of course, all this work, was done in vitro with purified proteins and so on. What about in vivo, and what about more complex proteins? Well, more recently, we've expanded this concept to the scale of designing enzymes. So here I'm showing you an example of the design of a particular enzyme called corsmate mutase. This enzyme works in a biosynthetic pathway in bacteria called the shikimate pathway that leads to the synthesis of the aromatic amino acids tyrosine and phenylalanine. And indeed, bacteria that are deficient in corsmate mutase are oxytrophic for these amino acids, meaning that they can't grow until you supplement these amino acids to them. This sets up a very beautiful functional assay to study both natural and designed corsmate mutases. So indeed, if you take a library of 1,000 or so natural corsmate mutases drawn from diverse species, and you put them into E. coli and ask if they work, here's the result. The data are on the x-axis show you the function of corsmate mutases in vivo, where 1 is the activity level of the E. coli enzyme, that is the natural solution. 0 is what you would get with just knocking out the gene, so this is like dead, right? And the bottom line is 40% of what nature makes in corsmate mutase homologs throughout the evolutionary divergence actually work in E. coli to an extent that's near what the E. coli enzyme does. And by the way, these ones that are not working, it's not like they're not corsmate mutases, they just apparently don't work in the genomic context of E. coli. So in some sense, this data set gives us a positive control for what we should expect for a design model to be able to do if it was perfect, right? And here I'm showing you a model that was made on these data, again, keeping just the first order and second order statistical terms of interactions of amino acids. And if you sample from this model, here are data of roughly 2,000 synthetic molecules that were sampled from this model. And with that kind of top hit sequence identity, we get synthetic molecules that rescue function in E. coli. And indeed, the distribution is basically indistinguishable statistically from what you get from natural homologs. So from these data, we would conclude that these sort of simple models called POTS models can indeed design synthetic enzymes with statistics that are essentially indistinguishable from the natural world. These enzymes are really enzymes. So obviously we purified and biochemically characterized some subset of these synthetic proteins. And indeed, the data show that these synthetic proteins show kinetic parameters of KCAT and KM and so on that are indistinguishable again from their natural, that of the natural enzymes. So, we conclude from this kind of work that with a relatively small set of numbers learned in these models, again, we're able to make synthetic enzymes that are indistinguishable from their natural counterparts, both in vitro and in vivo. And this really, the simplicity of these models really argues that there's just a lot of sequences in the sequence space that could be members of this protein family and work in a particular cellular context. And indeed, one of the beautiful things about POTS models is that you can compute the entropy of the model, meaning that you can compute the size of the sequence space from which you can sample that satisfies the constraints in the model. And that number is ridiculously large, right? So this is actually a lower bound on that number. So that what it says is that there are, at the minimum, 10 to the 25th sequences that work as corsate mutase enzymes, even in a particular genomic context. And that really tells you about the sort of the simplicity or the size of the sequence space in which evolution can operate to create these sort of enzyme molecules. Now, what's interesting about this number, of course, is that it's astronomically large, but also in some sense it's zero, because if you look at the size of the sequence space with no constraints applied at all, it's 10 to the 124th power, meaning that this is a space that you'll never find with any random search. But with models trained on these sort of statistics, we're able to reproducibly find sequences in that space. Think about this space as a kind of the space of alternative histories that could have happened in evolution but did not. And gives us, of course, a giant space to search for novel molecules. So indeed, in the past many years, there's been a kind of cottage industry of designing various molecules by these pure sequence-based models. Using various different approaches. Early on, it was these simple statistical physics models. More recently, with variational autoencoders, and more recently with large language models, which I'll tell you about in a second. The data show that we can make a plethora of different sort of proteins with various degrees of complexity and various interesting molecular phenotypes. Let me spend the rest of the time today telling you about the most recent model. Here's a new model architecture, which we have recently worked on. This is called BioM3. It stands for Biological Multimodal Model. And as the name suggests, this is now a model that's consuming different sort of data types into a coherent representation for improved protein design. So let me show you what the model basically looks like. So this is a unification of two large language models. So there's a language model that's trained on the entire protein universe. So the roughly 600 million or more sequences available in the uniprot. And so you can think about this language model as basically what evolution knows about protein structure and function. We then constructed a separate language model, which is now trained on the entire PubMed. So it's read all the papers in the scientific literature. So I think about this language model as sort of what people know about biology. And the interesting thing was then to create a joint embedding space in which vectors of the protein world are aligned with vectors of the text world by contrastive learning. There's some details about the way that this model is made that is worth getting into, but in the interest of time, I just want to make one point. The architecture that we've done here is interesting and different from other multimodal models in that we're looking at separate language models that are jointly unified in an embedding space. By doing that, one of the hypotheses is that we're cleaning the overfitting noise that's inherent in each of these models, because the coherent signal that's represented in the protein model and the text model is the only thing that's learned in the joint embedding space. And so in principle, that should be a purification of the true physical constraints on proteins and stripping away more of the sampling and bias noise that's inherent to each of these models. And I'll show you some data that supports that argument in a second. The bottom line is the output of this joint embedding space is a text embedding that then can get put into a new kind of autoregressive diffusion model to then produce a synthetic sequence. So the overall point of this kind of model is the ability to do now natural language prompted protein engineering. So let me show you the behavior of this model. So for example, in one manifestation, you can write a piece of English text and say, I want a protein that looks like X, and that text prompt then gets embedded in the latent space of this model in some particular address. And the diffusion model can then decode from that address and produce a synthetic protein. And what I'm showing you below here are an overlay of two structures. One is the synthetic protein produced by the diffusion model that's in color, and then actually a natural protein that lives at that address in the white color. And you can see that with very good statistics, the model is able to design synthetic structures that recapitulate diversity of protein structures in nature. And this, of course, is similar to what many people have shown, the ability of language models to really design protein structures. But the really interesting thing is about the design of function. So let me show you that result. So to look at the ability of BioM3 model to generate function, we focused on a beautiful test case, which is the SH3 domain family. So these are a family of small protein interaction modules that occur in many, many signaling pathways throughout eukaryotes. In yeast, actually, or in all fungi, there's a particular pathway mediated by the SHO1 receptor that responds to external osmotic stress and initiates a signaling process that mediates a cellular response to the osmotic pressure and is essential for growth under high osmotic stress. To just show you the non-trivial nature of this model system and a challenge for these language models to design, if you take a giant diversity of natural SH3 homologs and you put them into the SHO1 receptor pathway in yeast, and you look at the ability of these SH3 domains to complement function, the data are shown on the right here. So again, on the x-axis is a normalized score of function, where 1 means you work as well as the wild-type natural protein designed by nature in this pathway. And the bottom line is the vast majority of these natural homologs do not rescue. These are legitimate SH3 domains. They just don't work in this particular signaling context. But there are a set of 176 proteins that rescue, as well as the natural fungal protein. And indeed, those are all the orthologs of this particular SH3 domain throughout the diversity of fungi. And so that this really identifies a problem for these language models. There's exquisite specificity in the signaling pathway that really only about 2% of all the natural proteins will even work. So the question is, can text-prompted design make true synthetic orthologs of the SHO1 SH3 family? And indeed, here are the data. So we can write a text prompt that looks like what I show you here. We just simply say, I want an SH3 domain that interacts with this particular ligand. It works in this pathway that detects changes in external osmolarity, and it activates the signaling pathway. That's the text prompt given. And sure enough, the model produces synthetic sequences that we then back translate and clone into the appropriate receptor site that mediates this osmotic signaling pathway. And here's the functional response of these text-prompted signaling domains. So again, on the x-axis is the same normalized functional score, where 1 is like the natural protein, and 0 are just things that are dead. And indeed, what this shows you, and all the blue dots represent the text-prompted synthetic proteins. And indeed, the model is able to make beautiful sequences that work in this specific signaling pathway to an extent that mirrors what the natural protein can do. And indeed, we've biochemically purified some of these proteins, and we get binding affinities to the appropriate target ligand that are at or better than what the natural affinities are. What is different about this data set and this data set? It turns out that it's about prompt density. So the graph on the top is the result of using a heavy-duty text prompt that actually gives a lot of information, the protein name, the lineage, the specifics of the function, and BioM3 interprets that and makes a number of sequences with a fairly high yield makes functional sequences. But now the top hit sequence identity of these proteins to the nearest counterpart in the natural world is 78%. But if you strip down the prompt and ask a much more simple, vaguer prompt, I would say with less information, the model is still able to make functional proteins, but now with much greater diversity, right? So remember, this is an exponential space as you go down. So now we're able to make proteins with that kind of diversity that beautifully complement the signaling pathway. And the interesting result is that these proteins are now even more diverse than any of the natural proteins that complement this function from the natural world, okay? And I think this is sort of, for me, the first time I've seen a model that's able to make proteins with even greater diversity than what nature's made that works in a particular signaling pathway in yeast or in vivo. So, I think that's a really important result, but I will say one thing, that it's worth noting that the model doesn't really know anything directly about function, right? Which is really the x-axis of this plot. If the model really understood how to build function, it would build me sequences that were just in this red box, right? Things that were exclusively functional. But indeed, the model makes a lot of sequences that are also non-functional, right? So it's worth asking the question that what is the model missing that it doesn't know how to build proteins that are exclusively that are functional, right? So if you compare the sequence constraints that are in the functional proteins away from the non-functional proteins, the result is shown on this slide. These are two views of the protein structure of the SH3 domain. This is a surface representation, and this is a slice through the core of the protein. The red amino acids represent the positions that become invariant in going from the non-functional group to the functional group. In other words, the red positions are the motif the model should have put, right, in order to get this specific biochemical function of these particular subset of SH3 domains. Apparently, the model is not really aware enough to know to put this motif. This motif is interesting because it's not just amino acids that contact the ligand, but it involves an allosteric network that works through the protein core to distant surface sites. But this network of positions is something that the protein requires in order to be functional in this pathway. It's an interesting thing to point out that there are other evolutionary algorithms that can actually computationally predict these red networks. So one simple solution to generating a high yield of functional proteins is conditional generation with these motifs. And indeed, that's something that we are working on now. So to summarize this line of work, BioM3 is a multimodal model that in which we can do text-prompted engineering of proteins, where the text prompts can be engineered to control novelty of the protein sequence and to maintain the foldability of these molecules, and then conditional generation with these functional motifs that we can learn computationally allows us to make a very high yield of functional proteins, right? With no data-driven feedback tuning at all. Of course, data collection and reinforcement learning can be used to fine-tune the model to even increase the rate of finding functional proteins and in principle diverge the proteins beyond the limits of what nature can make. These are things that are upcoming in our work. Let me just conclude by saying that the BioM3 model is inherently scalable. One can envision training the model with additional data modalities. Indeed, the next one that we are going to add is protein dynamics. And I want to highlight one thing in this regard. The movie I showed you I show you here in the bottom right, looks like a molecular dynamics simulation, but this is indeed not a simulation. This is actually an experimental movie of a protein in operation in real time, in sort of the nanosecond timescale with atomic resolution. And if anyone here wants to know how to make movies of that sort, I refer you to these papers, which is a new technology to be able to directly do experimental dynamics on proteins. And indeed, such data are going to be critical to generate the core information to ultimately be able to make wholesale analyses of dynamics of proteins and to be able to include that into the modeling framework of these sort of multimodal models. So this is what we're looking in the future. I would say the next modality is in dynamics. And of course, everything I've told you today is about work at the molecular scale with generative models for proteins. But there's really nothing I told you about today that's specific to proteins. And indeed, data sets are available and growing at many scales of biology. And I actually believe that many of the principles we've learned here at the scale of proteins will hold at these higher level scales. So I'll just say the full stack of biology awaits, and I believe that these sort of generative models that we and others, as you know, including people that are speaking at this meeting or presenting, it's a very exciting future in which these sort of models might now really teach us the rules of biology at different scales and really enable proper engineering of these systems. So with that, let me stop and let me just credit the people in the group that have done this beautiful work. There's a number of really excellent students and fellows that have contributed to this work. Here's a list of our collaborators without which much of this would either not be possible or not nearly as much fun. And I want to thank the people that paid the bills. So let me stop there. And again, thank you to the organizers of this event for allowing us to present this work. And if anybody has questions, I know this is a virtual event, please feel free to reach out to me by email, and it'd be fun to discuss these results. Thank you again.
Thank you. Because this is a pre-recorded talk, so basically there's no question session. Although I'm really interested in the dynamic session, so I probably will email. And if you want to know more, and please email Rama. So the next speaker is Professor Pei Long Lu. Professor Lu is a principal investigator at the School of Life Science, Westlake University. His talk will be sharing insights on de novo design of functional transmembrane proteins. Welcome.
I would like to thank the organizer for inviting me to give a talk here, and thank Dr. Lee for the nice introduction. And I would also like to thank Rama because he introduced the two major keywords. One is function, and the other is dynamics. That's I'm going to talk about during my talk. So for de novo protein design, we are interested in using these trained AI models to generate new protein structures, sequences with new protein functions. And hopefully we can generate like new molecular tools, therapeutics, or diagnostics. And during this process, we hope to provide some new insights into biology. So this is what we are interested in. And last year, David won Nobel Prize for his contribution in computational protein design. And a fascinating designer protein world has been created. For example, this peptide, the scaffolds, these protein assemblies like nanocages, protein two-dimensional arrays, and other functional proteins like ligand binding enzyme or those protein-based binders. And what my lab is particularly interested in is to design membrane proteins. Membrane proteins represents over 1/4 of our human genome, and it represents more than half of modern drug targets. You can think about the importance of membrane protein because it lies right at the boundary of our living cells and our surrounding environment. So it will mediate the exchange of information, energy, and materials across the membrane barrier. And actually, membrane proteins can do everything. It can be enzymes, it can be receptors, transporters, all kinds of functions. And it can also be adapted to applications. For example, yesterday there's a wonderful talk talking about nanopore sequencing technology. The key component, nanopore, is a membrane protein. And also people have been engineering those channel rhodopsins to serve as a key molecular tool for optogenetics. And we are interested in achieving custom-defined functions through de novo protein design. That means this kind of function is never seen in nature. For example, here we show that we can design a transmembrane fluorescence activating protein specifically bind to an artificial dye and activate its fluorescence brightly on the cell membrane. And we need to define the structure and find out a unique sequence never exists in nature and can fold into this particular three-dimensional structure and bind to this specific dye. So when I was a postdoc in David's lab, we have designed these very stable and static transmembrane proteins, including these transmembrane dimers, tetramers, and also some functional transmembrane proteins like these pores, like including 12 helix channel and 16 helix nanopore. I want to emphasize here, so the function here is also static because we are creating some static pore on the membrane, so it reacts passively to the external stimuli, for example, voltage. So there will be a linear IV curve. So what we are interested in, we really want to design some dynamic transmembrane protein structures for sensing and responding to the stimuli. Because for our cells, we need to sense these stimuli and to make a response to better adapt the fast-changing environment. So for the first part of the talk, I would like to talk about the novel design of voltage-gated ion channels, which was carried out by 3 of my talented PhD students. So first, we learn from nature. So how does nature do this? There are voltage-gated cation channels. They use like voltage sensing domain. There's one helix enriched in arginines, so it can adopt conformational change and then transduce to the central pore domain to control its open and closure. There are also voltage-gated chloride channels. So it adopts a different topology. So it seems like a transporter. And the gating element here is a gating glutamate. And so it seems like we need a charge residue for gating. And we designed this 15 helix pentameric transmembrane channels. We choose the C5 symmetry because previously we have designed this C4 symmetry. It has no pore in the middle. And for the C6 channel, it has no gating property. So we wonder if this 15 helix pentameric transmembrane channel will be a scaffold for accommodating gating properties. And we kind of tune the parametric design pipeline a little bit to introduce a theta angle that represents a twist angle along the central symmetry axis, so we can make a funnel-shaped channel. And the next step is we introduce like 3 layers arginines inside the funnel-shaped scaffold. And these gating charges seems to play 2 roles. One is serving as a selectivity filter because it carries like positive charge. It should select for ions such as chloride, bromide. And the next key role is it can serve as the voltage sensor because you can imagine for these flexible charge, the side chains, it can react to the membrane potential applied across membrane, right? Because when the membrane potential change, it may adopt different conformations to regulate the diameter of the central pore to control its dilation. So interesting, when we test these designer channels in real experiment, it shows this exciting outward rectifying current. So for the green curve, you can see at the negative voltage, it is remain closed, very similar to the controls. But over 40 millivolts, it rapidly opens up. And it seems like in the AI world, so it's like a ReLU unit, right? So previously it remained closed, and then upon to a certain condition, it opens up. So this is crucial for sensing. I will talk about that later. And this is a raw trace. And we further validated it allows unidirectional influx of chloride into the cell. We can switch the ion conditions and to draw this conclusion. So we don't have time to go in details. And we also did single channel recording of this type of channel. So it shows a unitary single channel conductance at about 40 picosiemens. And the gating mechanism to voltage is that according to the voltage increase, so the open probability increased. At 30 millivolts, it barely opened a little bit, and then at 60 millivolts, so the open probability increased by about 6-fold. So that's the key mechanism for voltage gating. And we also solved the cryo-EM structure for our designer channel, which is a very high resolution. And if we compare the design model to our cryo-EM structure, these two structures can be aligned perfectly within 1 angstrom RMSD. And interesting enough, so we can identify like 2 rotamers adopted by the very top layer of arginine. So this arginine 157 kind of adopt 2 conformations. One we call like up conformation, as you can see here, and the other is down conformation, as you can see for here. So we can calculate the different pore diameter for these 2 distinct conformations and draw the conclusion the down state kind of represents the closed state, but the up state may represent an intermediate open state. And next we examined the conductivity for the DVGAC mutation variants. Basically, we make mutations for each arginine layer and replace it to a hydrophobic residue. And what we can see from the panel that if we mutate the very top layer of arginine, so it can abolish the channel activity. If you mutate the central layer, the arginine 161, so for the pink curve, so it seems like reduced a little bit, but the unidirectional conduction is abolished at very negative voltage. You can also observe it can permeate ions. If you mutate the very last bottom layer, so it didn't make any change to the IV curve. So I think the beauty for de novo protein design is that our designer protein should be tunable, not only for its ion selectivity, but also for its activation voltage. And we did some mutations to our designer channel and introduced a mutation which mutates the very bottom layer arginine to aspartic acid. And by adopting this mutation, the IV curve kind of shifted to the left by about 20 millivolts. So this is crucial because if you think about in our physiological condition, the action potential kind of peaked at roughly 40 millivolts. So that means our original version of design that probably won't work in real cells, because when the action potential peaked, it's just about to open, but then it's suddenly closed. But for this 20 millivolt one, this is different. Theoretically, it can open during the action potential and then allow the chloride ions rush into the cell to silence the neuron firing. So in collaboration with Professor Li Bo's lab at Westlake University, we kind of engineered this AAV virus and carrying our designed voltage-gated ion channel genes, and this can be injected into a particular neuron. It can be expressed there, and for different variants, we all observe the robust expression. And what's more interesting is that for the 20 millivolt opening channel, it nearly abolished the channel activity. So it only fares the initial 1 or 2 action potential events. After that, the neuron got totally silenced. But for the 40 millivolt open one, so it didn't change the firing rate by a lot. So so we draw this model. At 0 millivolts, our designer channel kind of represents a closed state. When the voltage became very positive for DVGAC, these arginines can adopt another different conformation to open the central pore to allow the chloride rush into the cell. And also we can tune the selectivity, tune the activation threshold by introducing mutations and further engineering to make it really useful in neuroscience study. And we can compare our designed voltage-gated ion channels against these native ones. So our DVGAC can adopt a much simpler and more stable structure. So this is our designer structure. These are native existing proteins. And also our designer VGAC possess unique ion selectivities. It is different from most ion channels. And also the selective filter is also different. They adopt a totally different mechanism. And the gating mechanism is also different. Native channels using calcium or this voltage-gated chloride channel, they use this gating glutamate as the voltage sensing element. But in our case, we use arginine. So this arginine can serve as an activation gate and allow the unidirectional ion flow. And also for the single channel conductance, it is also different from the native channel. So it is about 40-fold larger than the CLC channel. But it is similar to glycine receptor and GABA receptor, which are ligand-gated chloride channels in our brain. To summarize this part, so we have designed these voltage-induced conformational changes for constructing these voltage-gated ion channels, and they exhibit novel features for design. And also, it has unique ion selectivity and gating mechanisms. Remember, it can be tuned to adapt for neuroscience usage. It can modulate neuronal activity. So the next part of my talk, I'm going to talk about another story. So we have designed these transmembrane fluorescence activating proteins. So basically, we want to generate a fluorescent protein embedded in the membrane. You may ask why we need to do that. Because we need a better sensor for sensing the membrane potential or pH gradient across the membrane. So there are native proteins that can be engineered to be like voltage indicators or pH sensors. But the problem is that they are very faint in brightness, and sometimes they are not stable. So we hope that we can generate these de novo design proteins for any artificial dye. We can create an environment for binding to the water-soluble dyes within the hydrophobic membrane to introduce these artificial dyes to be bound within the membrane environment that for sensing for the voltage or pH gradient across membrane. And what's more important is that these proteins are genetically encodable, so it ensures the tissue or cell type specificity. So imagine we can, by using genetic approach, we can express this kind of protein on specific cells, and then we can add the artificial dye in and to monitor the change to those external stimuli. So this kind of hybrid probe may offer new possibilities for enhanced sensor performance. What we did is that we focused on a fluorogenic dye. That means in free solution, there's no fluorescence signal. Only when it binds to some proteins, it will become fluorescent. So we use this RIFDOC and the old parametric design generation to design a water-soluble protein first to bind to this, specifically to this dye. And then we took the AI networks that by using this hallucination procedure, by incorporating these amino acid compositions, and we constrain the design size to design the protein to be transmembrane protein. Because if we use our old version algorithm to design these transmembrane proteins, it will unfold and won't bind to the dye. So by integrating with AlphaFold network, we can make sure that the binding pocket during design is kept intact and would be fixed for binding to the dye. And it is remarkable, and we ordered like a few designs, and two of them like bind to the dye and can shine bright fluorescence, and after probably one round of direct evolution, it can outperform EGFP. And you can see this is the bright fluorescence activated by our protein. For the buffer within the, so in the presence of the dye, so there's no fluorescence signal. And the binding affinity, calculated binding affinity between the dye and our design protein is around 18 nanomolar for the best design. And So as I have mentioned, the brightness is better even than the widely used EGFP molecule. And the binding is also specific. So we designed against this specific HBC 599 molecule. But for this HBC 620, so this dye only differs by 2 atoms to the original HBC 599. So basically, these 2 small molecules are very similar. Our designer protein can also recognize this small molecule and activate the bright fluorescence for that dye. But if you change it to another similar molecule, so it differs by 3 atoms, but here there's single aromatic ring compared to a condensed ring here. But in this time, our designer protein won't recognize this small molecule, suggesting the ligand specificity is extremely high. And we solved the cryo-EM structure for the designer TM Fab. And clearly you can observe the ligand within the protein and nicely sits there and very similar to our design pose. So if we compute the RMSD between the design model and the cryo-EM structure, so it should be within about 1.5 angstrom. And we can express our designer protein on either E. coli cell membrane or mammalian cell membrane or Xenopus oocyte membrane. So our designer transmembrane Fab will nicely localize on the membrane fraction of either E. coli differences expressing strains and bind the fluorescent probe and activate bright fluorescent signal. And to summarize this part, so we can accurately design the ligand binding transmembrane protein. So for artificial dye, there's previously no binding protein. We can design such ligand binding transmembrane protein that specifically bind to this molecule. And so we have also developed a new method based on deep neural networks hallucination for design of transmembrane proteins. So the success rate is very high. So we only ordered about 6 designs, probably 2 of them worked perfectly. And the third take-home message is that TM Fabs are brighter than EGFP, which should be valuable for imaging and developing sensors. So we are now working very hard to generate like multi-color transmembrane fluorescent proteins and to test its application in imaging purpose or serving as different sensors. And also we can theoretically, we can place any water-soluble artificial molecule within the membrane to create a binding site to in a genetically encodable way. So why is that important? So this movie sort of inspired me a lot. So if you look closely into the structure of the TM Fab complex with a small molecule, so you can observe that the small molecule is almost totally encapsulated within the protein. So that means if the small molecule dye will go inside the protein, bind there, the protein must undergo a little bit unfolding or dynamics. So this inspired the third project. So we can, based on that, design a dynamic transporter. Because for transporter, you want to design such a molecular machine that can adopt different conformations, like outward facing and then occluded, bound with the ligand. And then it will undergo another conformational change to open to the inside of the cell to transport the substrate across the membrane. So it can be either unipolar or like antipolar. So here we just took an example that we are designing such unipolar. This driving force is mainly mediated by the chemical gradient force. So the idea is that we can design an occluded state, and then by thermodynamics, so driven by the concentration of the ligand. The protein will open to one side and then bound to the ligand, then release the substrate. So the key part is to keep the energy gap between the outward facing state to the occluded state comparable to that between inward facing state to the occluded state. Otherwise, it will bind there and then release away to the outside, so there will be no transport. So we did that by introducing a dual topology design. That means the C2 symmetry axis is parallel to the membrane. So the membrane protein can adopt like two conformations or the two distinct topology with the N-terminal on both sides of the membrane. So it is an anti-parallel dimer with the N-terminal on both sides of the membrane. So we can design the membrane protein to bind to an artificial dye and then design the transmembrane surface and to using AlphaFold 3 to make some validations. And the experiment results is exciting. So we can use a very simple in vitro transport assay by expressing the cells using different transporter designs. And then we can observe that the accumulation of the dye with inside of the E. coli cells expressing different designs. But for the nectar control expressing a C2 dimer, there's no pore in the middle, it won't accumulate this dye. So here's a real-time video showing the accumulation of the dye with inside the E. coli cells. And we were also able to solve the cryo-EM structure for two designs. One is shown here. You can see it adopts like dual topology conformation because we fused a soluble domain to one side of the membrane. If it's a C2 dimer with the symmetric axis parallel to the membrane, you will easily observe like two blobs on both sides of the membrane. So that can be solved clearly by cryo-EM structure. And also, if you compare the design structure to our design cryo-EM model, the similarity is very high. The C-alpha MSD is about 1.2 angstrom. And if you look at the central cavity for ligand binding, so we kind of worried in the, at first that this protein may be a non-specific pore. But in this case, you can clearly observe these side chains. It's definitely not a pore, non-specifically permeating our dyes. So there's structured cavity and which requires conformational change to allow the transport of the substrate dye. And by testing this idea, we choose different ligands. And as you can see from this plot, different colors represent different dyes. So by small modification to the dye or totally changed our dye molecule, so our designer transporter only selects for the original design target. So this is striking and suggests the ligand recognition and transport is specific. And we also made some mutations on the translocation pathway. And by introducing those mutations, the transport activity is almost totally abolished. And we also set up this kind of in vitro liposome efflux assay. And for both basically we are expressing our designer transporter on the liposome. And for empty vector, you can see the dye is kept with inside the liposome. And for loss of function mutants, according to different time course, you can observe that there's a little change of the dye. But for either the different designer transporters, at different time, so you can see the dye kind of be effluxed through the membrane, and you can see some empty liposomes. There's no fluorescent signal within that liposome. So to conclude this part, so we can accurately design these dynamic membrane transporters from scratch, and this transporters adopt like dual topology in membrane. So interestingly, if you think about the very origin, like nascent, like precursors for transporters during evolution. So these 3 helix transporters may serve as the precursor or ancestor, very old ancestor for modern transporters, as has been discussed by David Zhu, who is an expert in transporter field. But the nature, so in our nature, we cannot identify such 3 helix dual topology transporters. And it can transport a specific ligand. It's not a non-specific transporter. And there will be future applications. For example, we can express these proteins in cell factories, in fermentation. For example, if you are accumulating like very high value chemicals, so you won't let your accumulated substrate to inhibit their fermentation. You want them to be expelled to the medium to increase the yield. And also, it might be a way to control the drug delivery by specifically expressing these designer transporters on a specific cell type. And to summarize the whole thing, so we have been keen at de novo design of custom function transmembrane proteins with dynamic structures. So keep in mind, there are no similar proteins in nature by that way. So that's de novo protein design. And it can also provide some new insights into membrane biology. For example, for the de novo design DVGAX, it adopts totally different mechanism in terms of voltage sensing or ion selectivity over the native ones. And hopefully we can use these designer transmembrane proteins for a wide variety of applications. To conclude, I would like to thank our talented students at Westlake University and our colleagues for collaboration and funding support, and of course you, audience. And I would be happy to take any questions that you have. Thank you.
Thank you, Professor Lu. It's a very structure-based talk, very interesting, at least for me. Any questions? But I take the privilege to ask the first one. So for the first part of your presentation, you mentioned that the voltage gate ion channel on the R165, the 3rd layer, so you try to mutate it into different other residues, and then that eliminate it. That doesn't change the ion channel function. So do you know what is the mechanism for the 3rd layer of arginine?
Oh, thank you for the, it's a great question. So can you play the slides? So the 3rd, so we kind of guess that because we are introducing a aspartic acid within the pore. So that can slightly change the geometry or the charge state of the pore. And to make this subtle change to modulate the arginine conformations. So by doing that, we can control the voltage activation threshold. So it's kind of like play a counter charge residue that balance the membrane potential. So in that way, it can tune adjust the activation voltage. So that's our hypothesis.
Have you ever thought how to prove using experimental validations to prove that hypothesis? Yeah.
We are making more designs except for this aspartic mutation.
Thanks.
Thank you.
Please.
Hi, that was fantastic. I have a quick question. So this seems to be a completely or mainly a physics-based designing. So is it possible to sort of incorporate the evolution information in some way and to sort of augment what's being done here? Or yeah.
Yeah, great question. So I have been keep thinking about in collaborating with Fa Jieyuan, who's a large language protein model expert at Westlake University. I think for our structure-based design, we can really make like really stable protein structures or custom-defined functions. But in terms of the optimizing the protein's activity, I think because so it's still hard to consider the dynamics. So probably large language models can provide a different angle of view. Yeah, I'm definitely interested in that.
Thank you.
Yes.
Thank you for speech. I have a question is, do you have plans to design some more complex transmembrane proteins, such as some proteins suffer from misfolding because it's water unsolutable? And if we want to design such protein, can we make the water insoluble protein into the water soluble and will maintain its function? Thank you.
Oh, I thought your question would be, can we convert like membrane proteins into water-soluble proteins for like an easy study, right? Is that the question?
Yeah.
So there are literature actually published in Nature that people are using this AI system to water solubilize this, for example, GPCRs or ion channels. So there are reports on that. So that's a very nice paper, yeah.
Because I think the de novo design protein, if it's naturally existence, and the de novo design protein just be a better protein, and it just express naturally protein just can treat the disease. But if we can just express the de novo design protein, which can reach what the naturally protein cannot do, we maybe do more further thing.
Yeah, I totally agree. So that's why we do de novo protein design. Yeah,
Thank you, thank you.
Yeah.
So once again, we thanks for Professor Lu's brilliant talk, and we have
Thank
a
you.
short coffee break. And please come back at 3 p.m. Thank you.
In this era, what are the missions of a university? What actions should